Okay, thank you for your kindly introduction, and also I'd like to say thanks for the conference to give me this huge opportunity. And I'm a PhD student from the Korea University. So today I would like to share with you about the things I've been studying that the applicable effective culture conditions for a clinical trial, human pluripotent stem cells, and which mechanisms are involved to make it possible. All gathered here today, we fully recognize the value of the value and importance of human pluripotent stem cells, and also we know what we're doing and what we have to do. As you know, human embryonic stem cells and induced pluripotent stem cells hold great promise as a renewable source of cells for basic and applied research. And more, human pluripotent stem cell-derived cell types are important for application ranging from in vitro modeling to therapeutic use. They have revolutionized reproductive and regenerative medicine and by allowing the establishment of detailed molecular and therapeutic models for certain metabolic pathways and life-threatening disorders. They also offer significant contributions to genetics and pharmacology in designing and analyzing disease models that can be closer to in vivo than any other procedures available. Why are we doing this study constantly, intensively? We're working for the same goal. The final and ultimate goal is the clinical application using stem cells. No matter whether it's just cell transplantation and gene therapy or drug discovery, we have different strategy for the stem cell, but it's for the same reason. So many studies attempted in various ways for clinical approaches across the globe. But unfortunately, we have to overcome some limitations for these studies make it too feasible. I summarize roughly. First, one complication is that use of human embryonic stem cells require lifelong use of drugs to prevent immune rejection. Second, another disadvantage is that using human PSCs can produce tumors from rapid growth when injected into the patient. Third, the possibility of cells from human PSCs were genetically defective. If human PSCs were genetically unstable, so that transformed cells cannot be used successfully. Finally, many of the existing stem cell lines were grown with the necessary help of animal cells. It is unclear how many cell lines were developed with the safeguards in place. Among these limitations, we focus on the last one, the optimal culture conditions for human PSCs. So, for their full potential to be realized, both the growth of the undifferentiated cells and their derivatives must be performed in defined culture conditions. But defined gene-free and fetal-free systems have not been fully established for the realization of these applications since human ES cells were first derived and cultured on fetal mouse fibroblasts. So, defined cell culture system devoid of non-human substances and feeder cells would benefit human stem cell research and support the production of cells for future therapy. The currently culture conditions of human PSCs have some problems such as allogenic, genogenic contamination, and numerous codes and mass productions and batch-to-batch variation, things like that. In usual human PSC cultures, and researchers use spatial media and matrix, which are obtained from animal or synthetic complex mixture. Even if you've got a way to fully control the cells, it would be difficult to apply to be a clinical in reality. So that's why many researchers and global companies are trying to develop for optimal and safety and clinical grade culture conditions for human pluripotent stem cells. At the first place, in our previous study, we considered using human feeder for human embryonic stem cell culture instead of mouse feeder. We used the cells from they are originally placed in vivo. and use feeder cells from human placenta-derived cells. We have successfully culture of human ESC without exogenous basic fibroblast growth factor supplementation, which is well-known established master factor of human PSC propagation. Based on this research, we developed conditioned media from human placenta feeder cells. We call it PCCM from now on. And this idea was ruled out the unknown allogenic contamination that we've got initially concerned when we use feeder cells. Firstly, cells isolated from the chorionic villi of human placenta, and we produce conditioned media. And then we just did simple gelatin coating on the dish and culture human PSCs. It is incredibly easy and simple way because we know that human PSC maintain is a little tricky. In our currently study, we're successfully cultured without additional essential growth factors using only conditioned media obtained from the human placenta cells. In addition, cell adhesion and expansion are successfully succeeded tremendously, even without use of special matrix, like I said. We use teaser and matricial, the most widely used media and matrix for human PSC culture, as a control group of this whole experiment. They are long-term grows robustly and keep expressions of pluripotency markers such as OCT4 and OXOX2, and even do not lose their ability of differentiation to 3-germ layers in vitro and in vivo without any chromosome instability after long-term culture in PCCM. We checked through the karyotype and STR analysis. And then we are curious about this. How could it be possible? What makes it happen? What's happening inside there? To identify the factor, we performed a human cytokine array containing 120 cytokine captured antibody, captured antibody mobilized on membrane as shown. The membrane were incubated with control media and teaser and PCCM. Interestingly, 14 cytokine detected in PCCM, including GRO-alpha and IL-8, were significantly increased. And we examined for the actual quantity of major candidate cytokine using ELISA. As you can see here, the cytokine levels of basic FGF was high concentration only in teaser. We found only in teaser, and surprisingly, we found out the IL-8 and GRO-alpha bind with high affinity to CXCR2 receptor, chemokine receptor. Besides, PCCM contains all 7 ligands of CXCR2, and also we observed that highly activated CXCR2 on the cell surface compared to the control group. So we focus on the CXCR2. The chemokine receptor 2 activated reinforced senescence of somatic cells and induced apoptosis, and also associated with the aggressiveness and metastasis of several types of cancer cells, but its effects on human PSCs have not been fully verified. We hypothesize those factors required for human PSC propagation would act through CXCR2 receptor present on the human PSC surface. So we try to find out the role and mechanism of CXCR2 in defined medium from human for a human PSC culture. We want to know how it works. To demonstrate that fact, we've performed block to CXCR2 receptor on the cell surface using neutralizing antibody and specific antagonist. As you can see here, many colonies are differentiation greatly after 2 weeks treatment than the control group. We use 2 specific antagonists for CXCR2. They are significantly lose pluripotency and lead differentiated after 2 weeks treatment individually. The other way, we examine whether the repressed gene expression of CXCR2 has effect in human PSCs. We've tried to inhibit expression of CXCR2 using small interfering RNA system in all 5 human PSC line, significantly decrease of Stamnis marker according to the repressed CXCR2. and consequently, differentiation marker expression were increased. In order to investigate the correlation between CXCR2 suppression and ARC pathway in human PSCs, we performed Western blotting, which revealed a lower ARC phosphorylation level in the CXCR2 suppressed group than the control siRNA group. Moreover, the proliferative ability of human PSCs was dramatically decreased during this period in both groups. These phenomena are different from when we confirmed in control group. When blocked to CXCR2, including basic FGF conditions, cells were not fully differentiated and kept maintaining pluripotency. They are decreased proliferation rates, but sustaining pluripotency. At the first place, we predicted to CXCR2 or CXCR2 ligands are suitable substitute for basic FGF. But according to our observations, it seems quite different role. We assume that might be involving different pathway or independent pathway. So we had a look into the role of CXCR2 on human PSCs by block the downstream signal transduction or gene expression. And the CXCR2 ligands stimulates to CXCR2 receptor, which is secreted from human placenta-derived cells, and might be human PSCs regulated by those ligand receptor binding. But we're wondering what mechanisms inside there and regulate whole situation make it possible. We tried to figure it out and looked over considerably. We found out recently that some molecules are activated human PSCs grown in PCCM than the control group, which molecules are stimulated to mTOR. After that, we speculate might be CXCR2 involved in well-known mTOR signaling pathway. For more detailed investigations, we confirmed using more stable and consistent way to target genes suppressed by short hairpin RNA gene knockdown system. As a result, human PSA lines were extremely decreased of proliferation and stemness marker expression such as octopher nano and RAX1 after the infection. And when CXCR2 was suppressed, significant decrease of mTOR signaling molecule phosphorylation according to CXCR2 knockdown by siRNA or shRNA both. To confirm the role of mTOR in human PSCs, for 2 weeks treated rapamycin and R80-001, which are specific inhibitors of mTOR. As you can see here, human PSCs are differentiated and significantly lose their stemness and proliferation ability. Likewise, previous data when we blocked the CXCR2. And we examined what lineage led to differentiation when CXCR2 suppressed. We measured mRNA expression by quantitative real-time PCR 3-germ layer markers. As you can see here, see the data, CXCR2 suppressed human PSCs were mostly differentiated to endoderm and mesoderm lineages. Surprisingly, our data correlated with already published paper in 2009 by one group. They demonstrate that mTOR support the self-renewal of human embryonic stem cell, and also when mTOR block are suppressed in human ESCs, they observe lead to endoderm and mesoderm differentiation. But they did not investigate which molecules are involved in their pathway on the upstream or downstream. Above all, these data indicate that when cultured human PSCs without any additives, including basic FGF, regulated by different signaling pathway associated with stimulate CXCR2 on the cell surface, human PSCs depends on the CXCR2, their pluripotency and proliferation, and assume the CXCR2 involved with mTOR downstream signaling, which are important effects in human PSCs maintained regardless of the microenvironment. So this study identifies CXCR2 for the first time that support human PSCs proliferation and self-renewal. and provide a rationally designed humanized medium for mimicking the ex vivo culture conditions. Our result demonstrates that the role of CXCR2 and its related ligands as novel and crucial components for regulation of human PSC via CXCR2 mTOR signaling pathway. This is what we have studied until now. We will continue study for more detail in specific downstream signal transduction and how those are regulated, and are still ongoing, and we try to manipulate whole microenvironment to apply on clinical trial. I hope that in one day our study will give practical help to the patient who suffers from life-threatening disease all over the world. That's why we have to continue on our study. I would like to say thanks for for the members of Institute of Stem Cell Research, especially Professor Byung-Soo Kim is in charge of all this study, who is my supervisor, and thank you for your attention. Thank you.
Are you on to question?
Good job. A very nice presentation, really nice work. So there is CXCR2 receptor knockout mice. What is the phenotype of that? So sorry about that.
CXCR2 knockout mice, unfortunately, we didn't check that knockout mice because we use the human embryonic stem cell in human-induced pluripotent stem cells, so we do not examine in mouse models and mouse cells.
Okay, thank you. And before I give the microphone to somebody else, as an announcement for the audience, the poster session is going to be at 5 o'clock, where the lunchroom is. So if you have posters, please go ahead and put them up, because they will be reviewed exactly at 5 o'clock. Thank you.
Thank you.
So there are a lot of different tissues now that are being used to make iPS cells, blood, skin. Do you think your CXCR2 finding is going to apply generally to all different types, or might it be specific to one or another?
You mean including adult stem cell or somatic cells?
In terms of what the starting tissue is that the iPS cells are being developed from.
Yes.
Yeah.
But according to a lot of studies, CXCR2 acting different role in different cell types, different microenvironment. They acting senescence and apoptosis in somatic cells, terminally differentiated cells, and they involved in metastasis and aggressiveness of cancers. But embryonic stem cells and induced pluripotent stem cells, they support self-renewal and keep maintaining pluripotency. This is totally different story of the CXCR2 role. We speculate that. Yeah. Thank you.
Okay, thank you.
Thank you so much.